In the last class, I have explained the ABO compatibility and how to remember the compatibility of ABO and RH. I think you might have followed all those details in the last video. Today I am going to explain what exactly is this ABO RH and H substance and all. Even though this subject is very simple, it is little bit difficult to get a clear concept of that. So slowly, step by step, I will explain all these basics of this blood group system. As you all know, it is Carl Landsteiner who introduced this ABO system in 1901, and it is he who explained all these ABO systems, and later of course RH also, with the help of another professor. His name is Weiner. Anyway, that I have explained already in video number 1. You check 1M or 1E. 1M is in Malayalam and 1E is in English. So this is my second video. In this one I am going to explain exactly what are the basics of this OAB and AB systems. So our starting point is red blood cell. All of you know that there are around 5 million red blood cells in 1 cubic millimeter that is circulating in our system. 5 million in 1 cubic millimeter. And as you all know, the shape of the RBC is round, but its center portion is not having the same width of the periphery area. So it will be something like that. And if you look from the side, it looks like that. It looks like this if you look from the side. So actually it's very small cell, say 7 to 8 micron length, not length, it is diameter, and around 2 micron width in the periphery, and here it is only 1 micron. The thickness is lesser than this edge. So 7 to 8 micron diameter, 2 micron width in the periphery, and 1 micron at the center. This is the approximate size of one RBC, red blood cell, or red blood corpus cell. Both are same. So, if you look through an electron microscope, you can see so many antigens projecting from this area, different shapes, from all this area you will see antigens, different type of antigens, ANTGEN, antigen. These are all projecting from the surface of the red blood cell. This antigen is also called agglutinogen. So, there are two names for all these projections, which you can see only through electron microscope, cannot be seen with our naked eye. The cell itself is very, very small, and then these antigens are very smaller than the cells. Maybe there are more than 45 antigens like this in one cell, 45 types. So, that means actually there are more than 45 blood group systems of ABO. In ABO itself, not only in ABO, the whole surface of the antigen, there are more than 45. Some antigens are known as A, some antigens B, some antigens AB, in some antigens known as RH, or it is also known as D. In RH itself, there are apart from D, there are RH C, RH small c, RH capital E, RH small e, and it goes like that. Around 49 to 52 RH factors are there, different RH factors. What is the difference between this ABO system and RH system? First of all, you have to keep in your memory that this ABO system is carbohydrates. We can say sugar molecules arranged in different ways. That is the ABO system. ABO system is not only seen in red blood cell, ABO system is seen in other white blood cells, neutrophils, basophils, eosinophils, also in platelets, and you can see this in some other cells of the body also. Not only in cells, but also it is available in secretions such as saliva, tears, milk. This kind of secretions also contain this ABO system. Whereas RH is found only in red blood cells. Different kinds of RH. RH is not seen in any other area. Whereas ABO system sugars are seen in many areas of the body, not only in red blood cells, other cells, in GI tract cells also it is seen. It is not seen in one area, that is the CSF, cerebrospinal fluid. Cerebrospinal fluids. You know, the cerebrospinal fluid is coming from the brain. There are 4 ventricles from where it is produced, and it is coming down to cover our spinal cord. This details I will explain when that subject comes in another video. Anyway, keep in mind, carbohydrates or sugars are this group, ABO group. In the ABO group, it is seen not only in red blood cells, it is also seen in many other cells of our body, and it is sugar. Whereas RH is a protein. RH is seen only in erythrocyte or red blood cell. The medical name of red blood cell is erythrocyte. Erythro means red. So, RH is a protein, whereas ABO is sugar or carbohydrates. ABO is seen in many parts of our body except cerebrospinal fluid. These are seen, these antigens, or it is also called agglutinogens. Agglutinogens are seen on the surface of the red blood cell. There are more than 45 antigens seen on the surface of the erythrocyte. Out of these 45, Landsteiner noticed that ABO and RHD. RH are many types. Out of that, RHD and ABO system, these two systems are combined together, and it is used clinically in our hospitals and wherever necessary. And when a transfusion is done, they take care of ABO and RHD. The other RH, Small c, capital E, small e are not very important, but that is also considered when a serious analysis is done at the time of transfusion. Otherwise, they are neglected. So, RH is protein, ABO is sugar. ABO is found in red blood cell, white blood cell, platelets and all other cells, and also in secretions like tears, milk and the like. Okay. It is seen in feces and meconium also, and this ABO system, whereas RH system is only seen in red blood cells. So that is the introduction. Now we will go little bit more details. These are the people who explained all these things: Karl Landsteiner, Alfred de Castello and Adriano Sterli. These two guys were the students of Karl Landsteiner. He was the Austrian Vienna University professor, and these two students, they were the people who studied this subject and developed this ABO system. And I explained in the last video, Karl Landsteiner got his Nobel Prize in 1930 for his discovery. Also, Karl Landsteiner, with the help of Mr. Weiner, he explained the RH factor. So this was explained in the last video. Now we will go to the next details of A. So when we talk about ABO, I told you we have to consider the red blood cells, RBC. In this RBC, there is one oligosaccharide foundation found in this RBC. Oligosaccharide foundation. So suppose this is the oligosaccharide foundation. This oligosaccharide foundation means, oligo means few, say from 3 to 10. 3 to 10 molecules of sugar are joined together to make an oligosaccharide. So this is the oligosaccharide which is accumulated on the surface of the red blood cell. Oligosaccharide. Oligo means many, saccharide means sugar. So some sugars are joined like this together, and they are coming and accumulating on the surface of the red blood cell, oligosaccharide. So that is step number 1. So we have already noticed that oligosaccharides are accumulating on the surface of the red blood cell. Oligosaccharide means few sugar molecules combined together. So this is oligosaccharide. Oligosaccharide. That is step number 1. This is already there in the red blood cell. Now there is another molecule coming and attaching in this oligosaccharide. That is what is called fucose. Fucose means it is a cousin of galactose, we can say, because its formula is C6H12O5. One oxygen, if you remove from galactose, you will get fucose, and wonderful molecule. Normally the sugar formula is C6H12O6, as you all know. But here one oxygen is removed, so you will get fucose C6H12O5. This fucose is seen inside here in the liquid part of the erythrocytes. But this fucose, it cannot go and attach by itself. For example, I will show, suppose the fucose is coming and attaching here. I will put F. This is the fucose. So from inside, somebody has to help fucose to go and attach to this oligosaccharide. So this is fucose. This fucose cannot go and attach by itself to this oligosaccharide. He needs an enzyme to do this transfer job, transferring from this area to the surface, the oligosaccharide surface. So this job is done by an enzyme. You know, enzymes are doing many wonderful jobs in our body. So here also one enzyme is coming forward. From where it can come? It can come from the DNA molecule. A gene expresses, and thereby an enzyme is produced, and that enzyme comes to help this fucose, which was here, to be transferred to oligosaccharide. So the name of this fucose helper, or the enzyme which helps fucose to come and attach with the oligosaccharide, is fucosyl transferase. That is the name of the enzyme. This is expressed from a gene. This is expressed from a gene. That means gene is in the glucose in the nucleus. Here, this matured red blood cell, it doesn't have a nucleus. That is a peculiarity of this red blood cell. Then, if there is no nucleus, there is no enzyme fucosyl transferase. So, where is this nucleus? So, this nucleus was there when this RBC was in a development stage in our bone marrow. That is pro-erythroblasts. You know, this development of erythrocytes takes place in bone marrow, red blood cells. Not only red blood cells, all the cells are born in bone marrow. So, at the developmental stage, there was a nucleus. And from that nucleus, this fucosyl transferase enzyme was produced by the expression of that particular gene. And that gene is seen in chromosome number 19. Chromosome. The detailed study of this chromosome all will come in next classes. For the time being, you just remember there is a chromosome by number 19, and from that chromosome, in that gene, it expresses and fucosyl transferase is produced. It is this enzyme which helps for this fucose to go and attach to this oligosaccharide molecule. So, first step is oligosaccharide. The second one is addition of fucose to this oligosaccharide. You got the point? A gene expresses from chromosome number 19, and that gene is producing fucosyl transferase, and that fucosyl transferase enzyme transfers this fucose from this area to oligosaccharide and attach to this oligosaccharide. So, this oligosaccharide plus the fucose, this total substance is called H substance. So, H substance is seen in all cells, whether you are A positive, A negative, B positive, B negative, and all those areas you will see on the surface of your red blood cell this H substance. What is this H? Where does this H come from? This H means You know, these experiments were done by German and Austrian professors. Those scientists, they found oligosaccharide and fucose, this molecule, like a very thin film of mist, a very, very thin film of mist. That is, if you blow in a glass or a mirror, you will find a very thin film of water molecules. Like that, they observed a very thin film on the surface of the RBC, red blood cells. So they wanted to name that substance. You know, in German language, there is a word, H-A-U-N-C-H. This is a German word, haunch. Haunch means thin film. So they noticed a haunch on the surface of the red blood cells, and they called it H substance. That means haunch substance. But later, in our medical terminology, the doctors who came after, they were of the opinion that this H came from histology. Histo means tissue. So this H belongs to that histo. That is not the true story. The true story is this. Haunch is a German word. It means thin film. So they noticed a thin film of oligosaccharide plus fucose. So they named that substance as haunch substance, and in short they called it H substance. So that is H substance. It is found in the periphery or in the surface of red blood cells. It came from the word haunch. There is also an English word haunch. It means a projection, or in a human being or an animal, it is the loin area, the backside, the buttock area. That area is also called haunch in English. So the same spelling is there in the English word. That haunch means another meaning. In architecture, it is all a projected portion. So that haunch is not this haunch. This haunch is purely German, and this H came from this H. So this is the story of the foundation which is seen in the surface of these red blood cells. Now we will go to step number 3. Here the step number 1 was oligosaccharide. It is already seen. Step number 2 is addition of fucose in the oligosaccharide. So this combined molecule is called H substance. So this is number 1, oligosaccharide, and this is fucose is number 2, and oligosaccharide plus fucose is equal to H substance. Now we will go to the third step. Before going to the third step, please remember that this fucosyl transferase, which is an enzyme to transfer fucose to this oligosaccharide chain and attach it. This is from chromosome number 19. Just remember chromosome number 19, that gene is expressing and all this job is done. Now we will go to the next step. So again I will draw this. Here comes your oligosaccharide and here is your fucose. This is oligosaccharide and this is fucose. Now we will go to step number 3. In step number 3, there is a gene. This is gene number 9. Not gene number 9, chromosome number 9. This particular chromosome, it has a gene which expresses two jobs. Number 1, there is another subject which is coming along with this one that I will explain now, that is so that you will understand it clearly. Here, as you know, fucose is available here and that will be transferred. There are two more subjects, so two more substances here. Number 1 is N acetyl galactose amine. N acetyl galactose amine. That is one substance. Another substance is pure galactose. So these two substances are also available here, but they cannot go by themselves or transfer themselves to this area. So they need also helper. N acetyl galactose amine. From here it will come and attach to this area, N acetyl galactose amine. But it cannot go by itself. So chromosome number 9, those genes are helping. A gene expresses and an enzyme is produced which will take this N-acetyl galactosamine to this area. So, in that case, what is the name of that enzyme? It will be N-acetyl galactosamine transferase. That transferase, that enzyme is taking this N-acetyl galactosamine and attach to this H substance. N-acetyl galactosamine. We can say NAG, N-acetyl galactosamine. So, this N-acetyl galactosamine, with the help of the enzyme N-acetyl galactosamine transferase, it comes and attaches itself to this RH substance. So, now there are three things. This is oligosaccharide, this is fucose, this is N-acetyl galactosamine. These three substances together are called A antigen. So, what is A antigen? A antigen means oligosaccharide plus fucose plus N-acetyl galactosamine. When these three substances join together, it is called A antigen. So, if your blood group is A, that means you have oligosaccharide, you have fucose, and you have N-acetyl galactosamine. These three items together are called A antigen. So, in the red blood cell of all the A group persons, all these three substances are appearing on the surface of the red blood cell of that person. That is A. Now, what about B? Here I wrote another substance, galactose. Galactose only. Here it is acetyl galactosamine, that is another substance, and this is galactose alone. So, here again I am writing the oligosaccharide plus the F substance plus there is galactose. So, when galactose is transferred from this area with the help of galactose transferase or galactosyl transferase, the enzyme, the name of the enzyme is also very important. That enzyme is attached to the fundamental substance. So, you can easily identify that. So, here it is only galactose. It is all from the same gene appearing in chromosome number 9. A gene expresses, it produces two enzymes. One is N-acetyl-galactosamine transferase. Another is galactose transferase. Two enzymes. If so, this galactose is producing blood group B. So B antigen. So if a person has B antigen, that means his blood group is B. What does it mean? B. So if galactose is attached to this H substance, it is called B antigen. And the person is B group person. Now take another case. If this gene in chromosome number 9 expresses both these substances being transferred to here and here, then that means that person is having antigen A and antigen B together in his erythrocyte or in his RBC, as I have drawn here. In the left hand side, this N-acetyl-galactosamine is attached to the H substance. So this is A antigen. Here galactose is attached to the H substance. So this is B antigen. So A antigen and B antigen both are available on the surface of the red blood cell of AB person. Both are there. That is why he is called AB person, AB blood group person. Now think of another scenario where chromosome number 9 is not doing its job. That means there is no enzyme produced for NAG or no enzyme produced for galactose. Neither N-acetyl-galactosamine transferase nor galactose transferase. Both are not available. That means in that case only this H substance remains. No NAG, no galactose. That is one situation. I am writing here another H alone. So this will be H alone without any NAG, without any galactose. So this person is called O group. Actually, where is this O group coming? There is a German word Ohn, O-H-N E won't. This means without. The meaning is without. O-H-N-E. It's a German word. So actually, the scientists noticed that there is a situation where NAG is not there or galactose is not there. Only H substance without NAG and without galactose. That means without A or without B. None of them is coming. Only the H substance is remaining on the surface of the erythrocytes. H alone. That means chromosome number 9, this gene is not expressing these two substances. Actually, not these substances directly, the enzyme which transfers these substances to the H substance. The enzyme is not produced. So, there may be some mutation or some other problem in the gene, so that these enzymes for N-acetyl galactosamine or galactose, both enzymes are not produced. Only the H substance remains. So OHNE in German language means without. So H substance remains there without NAG, without galactose. That is why and how the O group came. That O means this O. But nowadays, doctors are just using this O in the sense that there is zero NAG, zero galactose, so that zero came and then zero became O. But actually that is not the story. It is from this OHNE, own, German word. This O is actually connected with the O group person. So O group person means without NAG or without galactose, or we can say without A and without B. OHNE is the word from where O group came up. Not from zero, then from zero to O. It's not like that. So A, B, AB and O. Now I think everything is clear to you. Is it necessary to repeat? Just I will tell you again. N-acetyl galactosamine, with the help of its enzyme, comes and attached to this H substance. Then it becomes group A. Galactose, when it comes with the help of its enzyme, comes here and attaches with the H substance, it is B. If both are coming and attaches this plus this in an erythrocyte, then that man is AB. When there is neither NAG nor galactose, that is nothing, then only H remains there. That means without NAG or without galactose or without A or without B. that is called ohne. So ohne means in German without. So without these substances, that is O group. So A group, B group, AB and O. Totally 4 important blood groups. And in the next video, I will explain the antibody. That's also a very important subject. Please wait for my next video. Thank you very much.